speaker is Professor Huang. He just joined UGA last fall. He is a assistant professor of biostatistics, and his research interests include biostatistics, medical imaging data analysis, and imaging genetics. He has developed several statistical and computational tools and applied those tools to monitor and detect the brain aging and dementia and other mental disorders. So I'm gonna turn it over to Dr. Huang. Today he's gonna talk about why statistical modeling means imaging heterogeneity.
Thank you. Nice introduction, and I think it's a great honor for me to have a chance and come to the IOB and present some of my recent research work with my students and also my collaborators here. And the title of today is when statistical model meets the imaging heterogeneity. So we can figure that I will talk about two different parts. So one is the statistical model and another is about the imaging heterogeneity. And actually, so today I think the talk is not specifically on one project. So I will just try to deliver several of my previously and also ongoing or some of the future works with you guys. So so during the talk, so you just feel free, and if you have any questions, you can just ask me. Okay. Okay. Okay, so in this talk, first I will talk about some background and the challenges, and then we'll move to some of work we have already done, and then we'll talk about some just in progress, what we are currently working on, and then we'll talk a little bit about the future work. So for the background and challenging, so actually with this rapid development of all this modern technology, so in these days, if you go to all these government agencies, you can see that they announce the different kind of large-scale medical imaging studies. For example, I just point out some of them, like the Human Connectome Projects, the Alzheimer's Disease Imaging Initiative, and the Osteoarthritis Initiative, the Cancer Imaging, the Archive, and also the UK Biobank. So some of them, maybe you are interested or you have some experience with these data sets. So if we take a look at these data sets, there are some very interesting characters over there. First, you can see that so in the recent years, during the number of the participants in the studies is getting larger and larger. And when I first jumped into this field as 10 years ago, so typically we can only recruit like 100, 200 patients or these volunteers. They want to work with us on these imaging studies. But recently, because there are a lot of money spent into these projects, now we can recruit so like this hundreds, thousands of patients into the studies. And another interesting is about this data set. So previously we can only connect like the imaging data and also some clinical variables from the patient. But recently, I think the goal of the different cohort of studies, they want to connect the different type of data sets, not only for the imaging or the clinical variables. They also want to investigate like this genomic data or other different kind of omic data sets. They also want to connect this kind of the epidemiology data set from all these patients or all these participants. And additionally, we also find that just focus on these imaging data sets, you can see that the people are just connecting different type of imaging data sets. We call this different modalities or multi-modality data sets. Like we have this MRI data, we have the CT image, or we also have some ultrasound image for different studies. It strongly depends on what is the goal of this study. Okay. So here I just want to spend some time to introduce some of this project or some of the data set or studies we are very familiar with, or we just worked on this in recent years. The first one is about the ADNI study. So ADNI study actually it has launched in the 2004, and it already takes around over 20 years. And it is a longitudinal, multi-site, and also multi-phase studies because, and for different time points and the research organizations, they have different research goals. For example, and for the adenine 1, which is the first phase in this study, and their goal is mainly focused on the develop this kind of AD-related biomarkers and from this imaging or some biological measurements. And then I think recently they just launched a new one, which is called adenine 4. So in adenine 4, so they are not only focused on this kind of AD study, they just want to try to explore or to generalize to some other different AD or this kind of aging-related dementias. Okay, so they start to recruit some patients that are not only as AD or MCI, which is a mild cognitive impairment. They just want to include some other different type of these dementias. So they have these new patients participants in the recent 2 years. Okay. And besides this kind of study plans, it also has this features that they just connect different type of data sets. For example, they have the different image modalities, including the MRI, the PET, or the diffusion tensor image. They also have the genetic data set and also this kind of biomarkers that is related to the Alzheimer's disease, like the CSF or some other different information. And another study I want to introduce here is about the OAI study. So this is not related to some of these mental health or neurodegeneration studies. That is about our disease. You know that when our getting older and older, especially for some specific subpopulation, like this kind of football players. So when they retire or just getting like 40 years old or 50 years old, they usually have this kind of concern about their knees. And because they have this kind of strong sports activities during their like the 20 years or 30 years old. And at that time, so typically, and all these patients, they need to go to the clinics. Some of them, they even need to take the replacement of their knee areas. So, which means this is a big problem for all these people when they're getting older. So in these OAI studies, they recruited around 5,000 subjects, and they collect different kind of data sets, like the demographic information. They also have this evaluation about the patient, like the pain evaluation, or this kind of socioeconomic status or medical records. They also have this kind of imaging data sets. They have the X-ray, they have this MRI data sets. So they just want to connect different kind of imaging data set, and then they can reconstruct these knee areas and evaluate this kind of status of these knee areas for different type of patients. Okay. And the next one is about the TCIA GBM study. Actually, the TCIA is mostly, I think some people they may get familiar with this kind of TCGA. So that is about this kind of the different cancer type research studies, and people connect this kind of genomic informations for all these patients. So TCIA is kind of this kind of parallel studies. They not only connect this genomic informations, they also connect this imaging data set from all these patients. So for this TCIA GBM studies, they mainly focus on this brain tumors disease. So typically, they connect this kind of raw MRI data, and for this raw MRI data, they have different modalities, like they have T1, T2, and FLAIR. These different kind of imaging modalities can reflect these tumor areas in the brain and accurately, and which can help these clinicians or these imaging readers to locate the tumor in our brain. Okay, and after they connect data set, they also have some clinical variables like age, gender, and the genomic information, and also you have the kind of survival status for all these patients. So typically, all these data sets are connected from this kind of cancer research centers like MD Anderson or this the Memorial Sloan, all these cancer centers, and they connect this data set together, and they build up this kind of research cohorts, which called TCIA and GBM. And once we have all this data set, typically we can just use some imaging processing and softwares to extract these tumor features, and which can be used for some downstream analysis, like the survival analysis, to predict how long and for the patients they can live, and also they can use to do some classifications to figure out the new coming patient is a cancer patient or just a normal controls. Okay. Now this is all these imaging studies. So depends on the different tasks or goals for these studies. And we usually just think about that, could we just use some statistical models to solve these problems? So when we talk about this kind of statistical modelings, usually we'll have this data set incoming, and then we always need to clean up this data set, because you know that when we connect the data sets, no matter it's clinical data set or imaging data set, usually it's raw data sets. So there's a lot of noise inside. We need to do all these data cleanings and to make sure that that data is ready for our modeling, or no matter what kind of model you want to do, you want to use a machine learning model or you want to use just a statistical model, okay? And also we need to do some this kind of data exploration, which means that we need to find some patterns in the big picture and to see that which model will be the best one for your problems. And after that, we can do these modelings, and then we can also use the model and to do some downstream analysis to evaluate some new coming subjects. Okay. If we apply this idea into this imaging studies, so typically we will get this kind of patient into this MRI or different kind of imaging scans. And after we get this data set, and we can just do some pre-processing, and then we will use all these data sets for your own projects. We do this data analysis, and then we can use different metrics to evaluate your models. It is a good one or bad one. And also we will ask some experts to help us to evaluate the models. If it's the good one, it can be applied to these clinical studies. And so here are some examples of the statistical modelings, and like the linear regression model. It depends on what kind of problems you want to solve. If you want to just, if you just want to investigate the association between the imagings and some clinical variables, you can use this linear regression model or multivariate analysis. If you are interested in this kind of predict this kind of survival status of your patients, you can use the survival analysis. But we always have these questions from your collaborators or from your communities that, okay, so we know the statistical models. Sometimes we see, this is too simple, but people may say that, okay, a simple is better because the model is simple, which means that you can do some statistical inference, which means that all the results is interpretability is higher than some black box models, right? But some people may say that, but I have more emphasis on this accuracy in these prediction problems, which means that, okay, the model is simple and can guarantee that the performance is also better. So now we have this kind of question, or we have this kind of discussions in these past several years. So people always have this debate that some people think about that, okay, we want to use a simple model like the statistical model or this traditional or standard statistical models. Or some people think that, okay, that is in the old school. We want to try some this kind of machine learning models that has a much better powerful tools in our imaging studies. And there is a new coming terminology which called is statistical learning. So what that means is that for this kind of traditional statistical models, so people usually give a lot of assumptions on your data sets, and people always use this kind of predefined models to do this data analysis. And in comparison, for the machine learning models, they don't want to apply any specific assumptions on the data sets, and they more focus on this accuracy, or they want more focus on these optimization problems in this whole framework. And for this, but the bad or this kind of drawbacks for the machine learning part is that, so typically, since this model is much more complex compared to these statistical models, so it is not that easy for them to do this kind of post-inference problems, but it is not that easy for them to interpret or to explain their results. Because of this, so people are thinking about, could we just to take all these advantages from the statistical model and the machine learning models and combine them together? So we call this kind of model is a statistical learning, which means that we want to give some fewer assumptions to a data set. We want to give some flexibility to the models, but we also want to keep these advantages of this kind of interpretability or explainability of our models. So this is the goal of statistical learning. But after that, this is not the key point in this talk. So we don't want to keep to discussion about which one is better. So the key point is that, so if you want to choose a model for n problems, it strongly depends on what the data or the complexity of your data set. So this kind of data complexity is usually happening in this imaging data. It's, for example, you may have this high dimensional data. You may have the data is not from this Euclidean space. For example, it is not some exactly numerical numbers. It's more like some data set from this non-Euclidean space. Or you have this kind of data has some heterogeneity here. So in this talk, we mainly focus on this kind of data with the heterogeneity. Okay. And when we talk about this kind of imaging heterogeneity, actually we have the different level or we have the different types of this imaging heterogeneity. The first one is because of pathological heterogeneity. So what is a pathological heterogeneity? So we know that when we get this kind of imaging data sets, so this kind of data may be connected from these patients. The patients, they have this kind of pathological patterns in their, can be observed in their images. So typically you can see that this kind of pathological patterns, or we call it abnormal regions, they usually significantly vary across different subjects in terms of their numbers, size, shape, and locations. So here is a very specific example. You can see this is the 2 MRI scans from the GBM patients. And if you take a look at this kind of tumor areas, which is segmented by some imaging reprocessing tools, the location and the size and the shapes are totally different between these two different patients. So we call this kind of heterogeneity is a pathological heterogeneity. Okay. And another heterogeneity I want to talk about today is this imaging heterogeneity. Is a study heterogeneity. So what is study heterogeneity? So you can see that when we connect this kind of large scale imaging studies, it usually connects data from the different sites, centers, or studies. So there is no one large scale imaging studies just connect from one single center or one single hospital. So if you do it in that way, you cannot connect like 100 or 1,000 subjects over there. It is impossible. So if we connect data from different centers or studies, and usually you will see that this kind of imaging phenotypes, they will show these distinct patterns, but this pattern is not caused by the biological patterns. This pattern is usually caused by this kind of difference in this environment or the population design or the protocols in the studies at different center. For example, some studies, they just connect the data set from this Asian area or have also another center is in the Europe or in the Africa. So you can see because we have a different population in each centers, and usually you will have different imaging patterns or some other different patterns in your data sets. So now we need to think about that. And we want to combine or integrate all this data from different centers together. We need to remove all these study heterogeneities from your data set. We only want to focus on this biological or this disease-related patterns in your imaging. Okay, so that's what we call the study heterogeneity. And here is an example. So you can think about that. This is the imaging data set that we observed here. And we also have all these factors. For example, we have demographic factors, we have the clinical factors, the disease status, study design, and the acquisition protocols, pre-processing, imaging processing pipelines, or some environmental factors. The question is, for all these factors here, we can only observe or we can only connect some of them. We cannot connect more of them, which means that some of these factors, they are observable, but some of them, they are hidden. So this gives us a big challenge here. If we want to investigate the association between these imaging phenotypes and some of these factors you are interested in, we can involve all these observed factors as a confounders. But the question is, we still have some, they are hidden. If you just ignore these hidden factors, so all this association analysis is not reliable. Okay? So that is the question. And another change is that some of these hidden variables, they are even correlated with these observables. So which means that sometimes if you detect this kind of significant association between your imaging phenotypes and some queries of interest, but this kind of significant results, it doesn't mean anything if there is any hidden factors associated with your factors, which that is cost, it's more like some causal inference. If you have some unobserved confounders over there, all this causal inference result is unreliable, okay? And that is the background and the challenges in this kind of imaging studies. So we talk about this model and we also talk about this imaging heterogeneities here. And the next part is about so what we have done in these previous years. So I think we will just talk about this different type of heterogeneities, and then for each heterogeneity cases, I will give some heterogeneity situations. I will give you guys some examples of these case studies here. The first one is about this pathological heterogeneity. So for the pathological heterogeneities, and we will talk about the first example is the TCGA GBM connection. So in this connection data sets, so we have over 100 subjects over there. And we also have this kind of pre-processed imaging data sets for all these subjects. And besides this imaging data sets, we have their gender and age information and some other like genetic information over there. And the scientific question here is, typically, we usually just use all these tumor area or imaging phenotypes to predict the survival status of each patient in these cancer studies. But in this, for this study, we are interested in that. So for all these tumor areas, all these imaging phenotypes, could we find any association between these tumor patterns and their biological information, like the age and gender? Okay. So typically, in these radiomics studies, so the straightforward way is people usually just try to use some feature extraction methods to extract the feature from these tumor areas, and then they will use this feature to conduct this kind of association with this variable you are interested. For example, here I give you guys two different ways. So the good thing is that this kind of feature-based approaches, they are very resistant to this tumor heterogeneity, which means that no matter this kind of size, shape, location of your tumor is, so we always can extract these features. And after we extract features, all these features are aligned across different subjects. And then we can do this kind of associations. But the question is, here we have two different ways. One is we call the radiomics feature. That is, we calculate all these features from these tumor areas. We can use this kind of intensity-based features, or we can use some texture features, or we can even use some advanced methods. For example, we can use some deep learning methods, and we can use these convolutional neural networks to extract all these features. Okay. And in this study, we just try this first way because this is very straightforward, and always people usually do it in this way. So we try to use this kind of this Python software, which is called the PyRadiomics. And this is developed by this researchers at Harvard University, and they want to extract some meaningful and useful texture features from these tumor areas. Or they can also, we also try to use this kind of pre-trained deep learning model, so VGG19. And we want to use this kind of pre-trained model to get some useful and important features from this deep learning model. But unfortunately, no matter for this kind of radiomics features, all these deep learning-based models, deep learning-based features, none of these features, we can find the significant correlations between these tumor features and this aging information. But actually, in all these existing studies, so people always find that, so this kind of tumor areas, or this kind of intensity of features, they are highly related to this patient aging information. So which means that this kind of traditional methods it doesn't work in our case. Okay. And to solve these problems, we have another different ways. So it's more like a statistical way. So which means that instead of we just extract some single values like this kind of feature from the tumor areas, we also can just try to extract some functions from these tumor areas. Like we always know that we can calculate the density function of all these intensities within these tumor areas, or we can just quantify these quantile functions from these areas. But the question is, the advantage for this method is that you can see that usually the function is better than a single number in terms of this information inside, right? If you give me a function, which means that I can not only give you this kind of that's a minimum value, the maximum value, I can also give you this kind of different kind of quantiles in your intensities among these tumor areas. So that is much more richer information inside this kind of function representations. But the question is, for some of these functions, if you want to use this kind of regression model to conduct these associations, this kind of function representations is not some simple functions, which means that they are from some non-Euclidean space or this manifold space. Because you can think about an easy way. Let's think about this kind of density functions. There is a very specific property for these density functions. That is, the density function, if you take the integral, it should be equal to 1, right? That is the basic property for these density functions. Now, if we just directly take this kind of density function as a response in your regression model, what is the problem? The problem is, if we just take one density function, you have another density function. You do this kind of simple calculation, and the result is never be a density function anymore. You can think about that f1 is a density function and f2 is another density function. You take this subtraction, f1 minus f2, nobody can guarantee this is density function. If you take the integral over there, the integral equal to 0, right? So which means that, so all these kind of function representations are not from this Euclidean space. They're from this kind of manifolds that is a nonlinear Hilbert space, which give us a lot of troubles when we want to conduct this kind of regression analysis. And here, we just want to introduce a new representation methods. We call it log quantile density function. After you apply this specific transformation, and then we can just transform this density function into another specific space. But this space, the good thing is that this is a linear functional space, which means that you can do any kind of summation, subtraction, and also you can do this kind of multiplication with some numbers. And no matter what kind of calculus you did here, and this kind of result is always there in this log quantile density function space, which means that, so for this kind of space, we can just apply this kind of functional regression model to investigate the oscillation between your imaging phenotypes and some biological variables here, okay? And here, after we just apply this specific transformation, that we call LQD transformation, and we can treat this kind of transformed functions as your response, and we can conduct this regression model here. But we want to give this kind of model some flexibility over there. So which means that we don't just want to apply this linear regression model. We want to apply this kind of semi-parametric model. We introduce this unknown non-parametric structure G function here, and we can learn all these potential nonlinear relationship between your imaging phenotypes and our queries of interest. Okay, so that is something different from these traditional statistical models. So we want to give this some uncertainty or this flexibility to your model. In that case, we can gain some advantages when we model this relationship between the imaging and these biological variables. And once we use our model and we do find this kind of significant relationship between the age and these tumor phenotypes. And besides that, we also find this strong or significant association between these tumor areas with this is this gender, and also we have this other different biological variables here. And besides this kind of inference results, our model is very powerful because we also can do these predictions. We can predict these density functions for these new coming subjects for their tumor areas over here. So this is, you can see here, this kind of blue line, the red line is observed intensities function for their tumor areas, and this red line is our predicted density functions for that tumor area. Okay. And also we build up this kind of software, which is based on the MATLAB. So it is already on the GitHub. Anyone, if they are interested, have his own tumor imaging data sets, they can just to dump this data set into this software, and you can conduct all this analysis I just showed you guys, like the association or the prediction problems over there. Okay. And now we move to the next case study. This is related to the OAI study about these knee areas. So in this study, we use all the subjects from this OAI study. In total, we have around 5,000 subjects over there. And here I just give you guys some background here. So the osteoarthritis disease actually is a common and it is also a chronic disease affecting all these joint positions. Not only for the knee, we also have the twist and all the different parts in our body. And if there is a joint over there, and it has this kind of risk to have this kind of OA disease. Okay. And in the US, there's over 50 million people, they have this doctor diagnosis, this osteoarthritis issues over there. And also, we also have this common sense as the OA symptoms, including the swelling, pain, and discomfort, the locking, and also other different problems in their joint areas. Okay. And typically, if we want to investigate this kind of OA disease, we really need to go to this kind of the cartilage areas between this kind of different bones in these joint areas. For example, and here is this for one patient, this is the knee. So this is a femur, this is a tibia, and we have these joint areas. So what we are interested in is not this kind of two bones here. We are interested in this kind of joint part. So this is, we call this different cartilages. The upper one is the femoral cartilage, and the lower one is the tibial cartilage. Okay, so this is for a normal control patient. You can see that this cartilage areas is very strong and the thickness seems normal. But if the patient has OA issues over there, you can see that their cartilage thickness is become very thinner, and which means that they have a strong risk to be an OA patient. Okay. And in this study, actually, we only have this kind of original data set from the MRI scans. But this kind of data set we cannot just directly use for our data analysis. We did a lot of this imaging pre-processing. For example, we need to extract this kind of 3D cartilages from this MRI data sets. And after that, we need to do this kind of imaging registration. And we also we want to make the problem easier, which means that if we always focus on the 3D cases, you can think about that. The 3D cartilage areas is more like some objects that is not that easy for us to do this analysis. What we are going to do is we only focus on the thickness of the cartilage areas. So we do this kind of projection from this 3D space to this 2D spaces. And then at each point, we have their thickness values at each voxel for this the cartilage area. Okay, so finally, what we are going to do is we only focus on this 2D thickness map for both the tibia and the femur. Okay. And the question here is, we have two different scientific questions. The first one is, we want to find there is some risk factors affect these cartilage areas. So like age, gender, or BMI, if some of them, they are highly correlated with our cartilage areas. The second scientific question is, so for different patients, we want to find their pathological patterns in these areas. But for the second questions, we face this very challenging problems. That is this spatial temporal heterogeneity. what is spatial temporal heterogeneity? So I want to talk about that. I think we have this. So this is not a baseline or this cross-sectional study. This is longitudinal studies. So we have this kind of imaging data for each patient at baseline, 12 months, 24 months, 36, 48, 72, and 69. So it's the longitudinal studies over there. And because of this longitudinal study, the question is, these pathological patterns, they have this kind of heterogeneity and two different directions. The first direction we call it the spatial heterogeneity. That means this kind of pathological patterns, they can be different across different subjects. And we also have this kind of temporal heterogeneity. Even you focus on one single subject, these pathological patterns can change across time points. At baseline, maybe it's still normal control, but after several different points, and then he or she may becomes an OA patient. Okay. And to address this challenge, so here we just to involve two different models here. The first one we call it is the dynamic functional mixed effect model, and compared to the traditionally mixed effect model, and we have to involve a latent variable here. This latent variable just help us to involve all these disease or pathological patterns into our model. But this is the latent variable, which means that we need to use another model to describe all these latent variables. So here we introduce another one. That is we call the dynamic conditional random field model. This dynamic conditional random field model is different from the traditionally conditional random field model because we have to capture this kind of pathological patterns in two different directions. One is the spatial direction, which means that, okay, so here we only, if we focus on this pathological patterns at this pixel, we know that this pattern, it can be determined by this neighborhood, all these disease status over there. But actually, we also know about that at the time point J, this kind of disease patterns or this pathological patterns is also affected by this previous time point. So we use this kind of model to capture this correlation, all these kind of heterogeneities from the two different directions. Okay. And after that, we just build up this whole framework for this study. We call it DADP, it's dynamic abnormality detection and progression. So we have this imaging analysis or imaging pre-processing. We get this kind of 2D thickness map from this 3D. And after that, we put this kind of 2D thickness map into our modeling, statistical modeling module. We can detect this kind of individual disease region detections. We can get all these pathological patterns for different subjects and also across different time points. And also we can get this kind of disease mapping for also this different subpopulations. We have this kind of, we can show that at each pixel of these cartilage areas, their corresponding risk probability to be a pathological disease patterns, okay? And also we can conduct this kind of association. For example, we can show this kind of p-values at each pixel and to see that if it is significant, affected by this kind of biological variables like the BMI. Okay. And that is two examples for this pathological and heterogeneity. And now I want to move to the next one that is about this study heterogeneity. So in the study heterogeneity, and what we are going to do is we have this data set from the ADDI study, but as what it says here, the ADDI study is a multi-site and also it's a multi-phase studies for different ways to connect the incoming recruited patients here. And in this study, we have all this data set is 798 patients, but not all of them are from a single phases. So we can see that around 600 from ADDI 1, the first phase, and then we also have some from the second and third phases. Okay. And for this data sets, our goal is we want to investigate this kind of association between these hippocampus areas with some variable we are interested in. That's age, gender, education level. So the question is, once we extract this kind of hippocampus areas from our brain, we can extract some features for these hippocampus areas. And what we are going to do is we want to do some data explorers on these hippocampus features. And we find that there is a distinct patterns of these hippocampus-based features across faces. Now you can see that if we see this context, the first context, second and third context of all these features in these studies, you can see that, for example, for this first context, the adenine 1 has totally different patterns compared to the adenine GO and adenine 2. If we go back to check this one, the third quantile and the adenine 1 and the adenine 2, they have different patterns with adenine GO. So now you can see that we have observed this kind of study heterogeneities in our data set. What we are going to do is we want to build up this, we call it hybrid factor regression model, which means that we want to involve all these variables we are interested into this regression model. We also want to take all these potential hidden variable or unobserved factors into our model as well. Okay, so in this model, you can see that we have this kind of X that is observed variables or factors. We also have this kind of Z that is unobserved ones. So the goal of this model is we want to simultaneously investigate the relationship between the Y, the imaging features, and all these variables we are interested in. And also we want to recover or to identify all these unobserved factors from our study. Okay. And here is the results. By using our methods, actually you can see that, so here the MVCM is some traditional methods. It didn't take this kind of unobserved or hidden factors into account in their models. But for our methods here, this is our methods. So compared to these traditional methods, we can observe that, so we can detect additionally or more significant associations between these hippocampus-based features and some queries we are interested. For example, education mass. So this one cannot be detected from these previously traditional methods. Okay, and also we can, besides this global association analysis, we can also do this local association analysis, which means that we can see that how this age or education level affects this kind of each local areas in this hippocampus. This is the left one, is the right one. You can see that this kind of association, they have totally different patterns across the left and the right hippocampus. Okay. And also, as we said here in our model, we also can detect or uncover all these potential hidden factors here. So this is what we find these kind of hidden factors. So we just do a simple association analysis between these hidden factors and all these observed factors in hand. You can see that it is very interesting. These hidden factors, they have some associations with this age information in our patients. And also you can see there is a strong study heterogeneity here from these hidden factors. I think this is the goal of our methods. We do want to handle these potential study heterogeneities in our data analysis. Okay. And that is what we have done in the past several years, and what is in progress. And so now I want to talk something more beyond this image heterogeneity. That is what we are going to do, what we are doing right now. So one is about imaging genetics. So for the imaging genetics, I think we started to work on this part. That is like 5 to 6 years ago. But at that time, most people are working on these kind of association studies, like this kind of GWAS or some other different kind of TWAS or this the EWAS. So what we are going to do is we have this kind of genetic marker. We have the environmental factors. What we want to do is different from this traditionally associated analysis. We just use this kind of imaging phenotypes extract from our brain imagings. We treat them as these phenotypes. We want to do this kind of association and find this kind of genetic variations in our brain associated with some disease. Okay. And this is what we developed in this in the previous years about this software. We call it the fast voxel-wide GWAS pipeline. And we can extract some imaging phenotypes from our brain imaging, and then we treat them as a phenotype, and we conduct this voxel-wide GWAS analysis, and we can get all these top SNPs over there and find some potential genes related to this disease. And recently, because we know that's the success of this causal inference, and now we are thinking about maybe association is not enough. We need to go further beyond this association. So what we are doing is we want to talk about this kind of mediation analysis, because in practice, we not only have this kind of environmental or genetic markers, we have this brain development and imaging studies. We also have this kind of mental illness status for all these patients in hand. So which means that, so typically we know here we have this kind of direct effect from these genetic markers through this mental illness, but we also find some potential pathways, that is the genetic markers, they will affect our brain structures, and then they'll consequently affect this kind of patient mental health status. Okay, so this is what we are going to do is we want to identify these potential pathways from these genetic markers to this mental health status through our brain structures. Okay. And in the existing studies, so we can see that we have a different kind in this kind of imaging genetics, we have different kind of genetic markers like the candidate SNPs or candidate genes. We also have the genome-wide SNPs. We have this kind of genome-wide genes. And for the imaging mediators, we have this kind of the region summarized statistics. We have this subcortical surface or this white matters in phenotypes, or we also have some connectivity-based mediators, like this kind of matrix type mediators. But if we want to conduct this imaging genetics from the association to mediation, and we have several limitations or challenges over there. The first one is, as we know, if we just focus on this kind of genome-wide SNPs data sets, so you already have this millions of SNPs over there. So we need to handle this high dimensionality issues over there. And also you will see that this mediators is not as single values anymore. So we have this imaging-based mediators. They usually come from this very different space. They have a very complex structures over there. And lastly, for this association analysis or for this mediation analysis, there is one important tool is we need to do this inference. So we just say to more straightforwardly is that we want to get some p-values, right? Once we have the p-values, and I can tell you that, so which SNP is a significant one, which one is not, right? So in this study, and we want to focus on an important region in our brain, which is the corpus callosum. This is the largest white matter regions in our brain. It's just in the middle. It's have this very important rules to connect our left hemisphere and the right hemispheres. And you can commute all this information from left to right and from right to left. But if we just focus on this kind of study and we treat this kind of mediator as some information from the corpus callosum, for example, we can use this volumetric information from these regions as the mediators to conduct this mediation analysis. The question is, if we just use this kind of CC volumetric information and we do this kind of analysis, so unfortunately, we cannot identify any pathways from the genetic markers to this clinical outcomes through this CC area. Now we need to think about that. So sometimes if you cannot identify this kind of pathways, so it may be caused by you didn't choose these mediators in the right way. So now we are thinking about that. Maybe we can just think about some other different representations from these CC areas. So we develop these methods we call the SGMAS. So this is stand for this shape-based genome-wise mediation analysis studies, which means that instead of just extract some volumetric information from these CC areas, now we are more focused on the shape of these regions. For example, if some AD patients, we usually observe this kind of an atrophy in these CC areas. So it cannot change this volume of these CC areas. It can also change the shape of these CC areas, okay? So if we use this kind of CC shapes over there, and then we conduct this whole framework, so we can identify 21 SNPs. They have a significant effect on these CC areas. And now we just go further to detect this kind of potential pathways between the genetic markers to the clinical outcomes through these CC shapes. And we find that there are 6 SNPs. They have this kind of significant pathways from these CC shapes. So here we also can get this local analysis, find this kind of local areas that is significant in these pathways. Okay, but for this analysis, we only focus, we use this kind of clinical outcomes is this ADAS11. That is questionnaire typically used to evaluate this kind of the neurocognitive performance for all the patients. And we also try to use another clinical outcomes, that is CDR-SB. So what is the difference with the CDR-SB and the one we used in the previous slides? So the CDR-SB is more likely to be used if we want to do some early detection, like the patient is at this MCI steps, not go to the AD patients. So we can see that compared to these previously findings, we find some additionally genetic markers here that has not been discovered in these previous slides. And also for some of these detected SNPs, we compare their overlaps of these identified pathways. So we find there is some common pathways for both this ADAS11 and also the CDR-SB clinical outcomes. But something is interesting. You can see that. So for the CDR-SB, so usually, so this kind of pair of these CC areas, they have been found to be significant in these pathways. But for the ADAS11, both the head and tail of the CC areas, they are playing very important roles in these pathways. So we have this kind of statements that, so maybe at the early stage of this neurodegenerative disease, like this kind of MCI stage, so this kind of AD or this genetic effect will first affect this hair part of the CC areas. And when the patient is getting older or just go to this late stage of this Alzheimer's disease, and then they will have this kind of effect on both the hair and the head. So this is our interesting findings by using the different clinical outcomes. And all these results, so we have some papers over there, and so some of them are still under review. So if you are interested, and you can just go there, or you can just email me, I can just share it with you. And the next part we want to talk about is, I think it's the most hot topics in recent years about the large language model. And we spend a lot of, I think, after I joined at UGA and I talked with some faculties here and who has a lot of experience for this large language model, and we try to think about that. And could we just to use the large language models into these AD studies? So what we have done is that, you know that in all these existing studies, so different groups or the different studies, they have discovered the different findings between the AD, the brain regions, and some genetic markers. But the question is, could we just build up this kind of systems to have this, the AD researchers in this community, they can just to find out all these potential connections or relations among the threes using one platform. What we are going to do is, we build up this kind of large language model based platform. We call it ADGPT. In the ADGPT, actually, we will just use this existing database, like we have this the GTEX. We also have the OMIM. That is all this biological database, and including all these existing findings in this biological community. And we extract some key informations from all these publications, like all this information related to the AD brain regions and some genetic informations. And we will just focus on 4 different tasks here. We want to identify some basic information of the genes. We want to identify this kind of gene, the relationship between the gene and Alzheimer's disease. We want to identify this kind of relationship between this gene and these brain regions. And also, lastly, we want to identify the relationship among the 3, the gene, Alzheimer's disease, and brain regions. Okay. And what we are going to do is, we will first use some pre-trained large language models, like this kind of llama or BERT, and we use this kind of models, and given this based on our constructed database, and we can build up this kind of ADGPT. And after that, we need to do some fine tunings to make sure that our models can better fit our own data set. And we also compare with some popular large language models. So you can see that here for the different tasks, we compare with all these existing popular models, even including like the DeepSeek, which is very popular in the recent large language model community. And we also have this kind of Grok 3.0. This is the one developed by this XAI, which is the company by this Elon Musk. And you can see that for no matter what kind of tasks, we always outperform the other different large language models in our performance comparison. And here is highlighting our methods. And now I want to move to this future work. I think the future work has 2 different parts. And the first one is we want to go further in this large language model parts. And for example, in our imaging studies, as we just mentioned over there, we have different data modalities. We not only have this imaging data set, we also have this clinical variable. We have this kind of genetic information. So the question is, can we just try to build up this kind of platform based on this large language model, and we can integrate all this data set together and to help us to do this kind of prediction of AD, or even just like this kind of information retrieval, like what we did right now, okay? And the next part is about this COVID and brain mental health pathway. And recently we have got a chance to access the data set, which is called ABCD. And in this data set, it is very interesting because first, this is longitudinal data sets, and they connect all these patients is between 80 years old to 12 years old. And all the patients, since this is longitudinal studies, it started before the pandemic and it's still going. So which means that we have all this patient information before pandemic and after pandemic. And also some of them, they have this kind of COVID-19 during this period. So what we are going to do is we want to investigate how this kind of COVID-19 affect their mental health illness. And also we want to identify this kind of pathways through their brains, okay? So this is a very interesting topics I think especially in these days. Okay, and finally, and I want to acknowledge to some of my students, and some of them are just coming to UGA, and some of our students as the Florida State, which is I previously stay over there. And some of students, they have already graduates, and the first one, the second one, they are the primary authors in these publications I just discussed in this talk. And now they are postdocs at Yale and the University of Tennessee. And also I want to thank to some of my collaborators here. And so I think now I just want to talk a little bit about this kind of student mentoring or training. So I think according to my previous experience, so because this is like this kind of imaging study-based lab, so we usually recruit students in our lab, and then we'll spend a lot of time to help them to understand these basic ideas or elements in these imaging studies. For example, what is the imaging modalities? And for different imaging modalities, how you use this imaging pre-processing software to process these data sets. And also we have some existing pipelines or existing softwares over there. And we will just teach the students and how to use our own pipeline or softwares to do this data analysis. And that is what we have already done in the past years. And of course, for the students' own projects, so we typically have two different ways. One is more focused on this methodology part, because we need to teach the students and how to develop their own methods, especially statistical modeling parts, and to model data sets, to interpret the data set. They are more AI-based, and we have all these large language model-based methods. We have this kind of deep learning or architectures over there, and you need to know how to build up this kind of deep learning methods and to solve these problems. And it's maybe just like this kind of challenges in the different communities in these days. We have the different cargo challenge or some other different grand challenges over there. And sometimes they just assign this challenge to some students, and they will just make a team right now, like 2 or 3 students. They'll work on one challenge and then try to build up this kind of deep learning based methods and to solve their problems. And we never want to, we want to achieve this kind of the champion in that challenges, but they know that it is very hard. So we just want to help students to get the experience and how to get involved in these challenges and how to build up these methods in such a short time period. Okay. Okay, so I think that is all for today's talk, and thank you very much for your attention, and I'm very welcome to any questions here. Thank you.
So what is it? DeepSeek.
Yes.
And so this is R1.
Yes. And it's actually, I think it's we didn't use that one because someone is we need to pay for it.
Yeah.
So what we are going to do is here we use the DeepSeek, which is distilled from this Llama 3.1. Okay. Yeah, this is what we use. We just do all this location at our own laptop, local environments, and then we try to do the same tasks. We just do it for our ADGPT, and we do the comparison here.
I was just going to say that, like, for a lot of these bigger models, like they usually give early access to academics. Were you given like an early access API key or something? Okay.
Yes,
cool.
yes.
Because I was going to say, like, this came out like a few weeks ago. In fact, you've already benchmarked, fine-tuned it, and everything. It's just like crazy fast. I'm like, holy crap.
Yes. Yeah, we spent I think students spend a lot of time on this part because. And for some of the tasks, it's not just the answer is not yes or no. So which means that if we want to evaluate our methods, we cannot just use some traditional metric like the accuracy to evaluate them. So we also ask a lot of students to help us to build up this metric, and they were just to do this evaluation on different tasks and different questions over there. And finally, they can help us to give this final performance result. Yeah. Oh, thank you very much. Thank you.
Any question? I've got a question. Nope, missed it.